So today on the show, I have Dr. Aaron Gonshor. He is a PhD physiologist and a maxillofacial surgeon. He has published and lectured extensively on implant therapy and the use of stem cell technology in tissue grafting. For over 14 years, he headed the clinical advisory and educational boards of both a surgical and surgical science systems providers of surgical equipment and expertise in the fast-growing area of salivary diagnostics, which led him to create Fluids IQ. And now he lectures extensively on the biological foundations of intestinal permeability and the salivary hormone analysis. And a big welcome to Dr. Aron Gonshor. You've already heard all about his bio, and now he's going to tell us a little bit more about himself, as well as what led him to create Fluids IQ.
Well, hello, Lisa, and to your audience, thank you very much for having me. I'm really very, very excited about spending time with you. My story is an interesting one, I think. I started off as a PhD physiologist, as a scientist, and moved from there into a professional career where I entered into maxillofacial surgery. I actually spent quite a few decades doing reconstruction of jaws and faces and taking bones from hips and doing all kinds of other damage, and never left my love of research, which had to do with the oral area. I became specialized in the area of saliva in terms of a lot of research we were doing. And so a good number of years ago decided that I'd like to really go back to my first love, which was research in physiology, and opened up a lab which specialized in saliva. And we did that in Toronto, and it is an ISO accredited laboratory. ISO is an international standard that is very difficult to get, and most labs in the world, if they want to be part of the upper level club where other labs will talk to them and so on, they need to attain what's called ISO 15189, which we did get a good number of years ago. For those who are from the United States, that's sort of equivalent to what's called CLIA. And so these are high standard labs. And our area of expertise was saliva. And we chose hormones to be treated with the medium of saliva, as far as laboratory work was concerned. And so that's what we started to do. And so we were doing adrenal hormone checking. We can go into that in a few minutes, and sex hormones, etc., because saliva is known as really one of the gold standards for looking at hormone levels and so on in the body, which we can go into in more detail. And we have continued to do that, but it didn't take very long before, by some stroke of luck, we happened to find out that we had the good fortune to be able to start looking at a peptide called zonulin, which again we'll discuss in a few minutes. And that led us to opening up a series of tests, which we call our Intestinal IQ series. And that is looking at biomarkers for intestinal function. And we used as the platform for that blood spot, which many of the people listening would know that if you have little babies, little preemies, and you want to be able to take blood, you take it from their toes or their feet, and you actually have them drop some blood onto a card. The nurses do. Or diabetics will quite often take some blood from their fingertips and place it onto a card. So they have a blood spot. And when that dries, it's called a dry blood spot. And that is what you send to the lab. So that really allowed people to be able to do testing, just like with saliva, in the comfort of their homes, and then to send it out to the lab. And in the lab, we would do whatever labs do, the magic they create to be able to then do whatever they need to do. So we did, our second group was intestinal IQ, blood spot testing for Oh, there it
Just
is.
just
There's,
for those of you watching on YouTube, because this is actually on my list for today, as well as what the other tests that we're going to talk about too, because I'm doing them all.
and I'm just going to tell you that the last thing that we did just a little while ago,
Mhm.
it's actually last month, we introduced our first series of tests using dried urine. The
Oh,
same way that you do dried blood spot, you can now
yeah.
do dried urine,
Mhm.
which is even easier because you don't have to poke your finger. You literally have to have some urine in a glass, and
Yeah.
to put the card looks very much like the dried blood spot card, and you just get the urine to saturate the card, let it dry,
And
and
there you go.
again, comfort of your home,
Yeah.
and you send it out to the lab. And that we call our metabolic IQ. It's metabolic wellness profile. Yes, and it comes in an envelope. Exactly. It's
Just like
really easy, very easy. And we'll go over, I don't want to steal our thunder for the next few minutes, but that is really dealing with metabolic function, which is another
yes.
key thing that we need to do. And I need to state right in the beginning that right from the get-go, our lab designated itself as a lab that does wellness testing. These are non-diagnostic tests.
Yes.
They're extremely important.
Mhm.
They're screening tests. They give you tremendous amounts of information, but they're not diagnostic. That means that both the healthcare professional and the patient or client need to understand that the healthcare professional that's dealing with them is not doing what a physician would do, which is to
Right.
do diagnostic tests. None of these tests tell you, oh, you have celiac disease, or oh, you have Addison's disease, etc. No
Yeah.
such thing. It will give you a roadmap and help you understand more of what you need to do to eventually get at that answer, which will include other healthcare professionals,
Mhm.
including physicians, etc. But these are wellness, non-diagnostic screening tests.
Yes. Yeah. And again,
How's that?
as you stated, very important. And so I want to dive into gut health. And it's been said many, many times that our gut is our second brain, and without our gut functioning well and absorbing well and getting our nutrients into our system, then we're not going to be well. And so let's get into that, and then we'll talk about how zonulin plays a big part in that.
Well, it's only been recently, during the time of Socrates, you know, during the Greek time, that said that the gut is the foundation of all our health. They didn't quite understand all the ways, but clearly they understood how important the gut is.
Yeah.
And it doesn't take much to really understand why that's true. I mean, the gut is, in fact, that tube that starts in the mouth and ends in the other end,
Mhm.
uh, is really, if you think about it, an external tube that sits in our body.
Yeah.
And we're pushing stuff down that tube constantly, liquids and various solids, etc. And the body has to figure out a way that it is going to be able to take whatever is coming in there, if it's nutrients, whatever it is, and make sense of it,
Mhm.
take what it needs, not allow what it doesn't need to go into places that it shouldn't be, and at the same time to be able to signal to the rest of the body that everything is fine, or if it's not fine, what do we need to do to stop the trouble before it gets worse?
Right.
And that's asking a tremendous amount of what is in fact our gastrointestinal system. And it is a beauty to behold the way in which it is able to function. On top of which, it is the home of somewhere in the area of about 150 trillion bacteria. And so when you think about it, bacteria make up, and it's called the microbiome. For those who are listening, you know that that's now something everybody hears about. Like, what
Yeah.
is this thing, this microbiome? And in fact, it is a wonder. It is all of these bacteria in their myriad colonies living within the gastrointestinal system that are not only there to help us digest, to help us keep immune balance, to keep us healthy, but in fact, they are absolutely necessary to our being alive. Very clearly, if you don't have bacteria in the gut, you're dead.
Yeah.
You can't stay alive because they are absolutely essential to breaking down many of the nutrients which we have to have in order to be able to live. And so, thanks to the bacteria, we're able to do that. And so, that's also in this tube that is sitting in our body. So, yes, the gut is absolutely essential to the health of our body, and we need to understand how it works. And the good news is that in the last several years, hasn't been that long, we've begun to get a much better idea and are solidifying our understanding of what the gut does for the rest of the body. But make no mistake, we're only at the beginning. We're making baby steps in understanding how it works. And what we're seeing is absolutely astounding. And so we can get into that.
Yes. And so zonulin, right? Peptide. And more. I watched a video with you. I was doing my research, and I can't wait to watch a couple of more, because you got into autoimmune, which we're going to get into a little bit today as well. And it was astonishing, the reestablishment, the modulation. We're going to get into that, and just with the effects that produced and brought to the table and what you've discovered. And so let's share zonulin and how that all fits into that picture.
Well, before you can discuss zonulin, what you really need to understand is how we now understand that the gut actually works. More specifically, what is the function of this one cell thick, just one cell barrier, which represents the intestinal barrier of cells that do all these wonderful things. And this one cell barrier used to be considered like a mortar and cement in a home. You had bricks, you had cement between the bricks, and that's what closed off the area. And somehow the cells managed to get stuff, nutrients through those cells, and it went to the other side. And that's the way you created the energy that you needed, etc. Well, that's not the way it works. In fact, going back about 50 years ago, there were a good number of studies that came out that said, you know what, it's not mortar and brick. In fact, it's just the opposite. That what we do have is a very dynamic system. Not only does the cell do its own thing and transports little nutrient pieces through it, but between the cells, which used to be the mortar between the brick, in fact, it's anything but mortar.
Right.
It, in fact, is a series of doors that open and close, and those doors allow various types of nutrients and other things to enter, and things that the body can get rid of, that it can push into the gut if it doesn't need them anymore, or it says, I don't want you in my body, I want you out into the gut so you can eliminate it. Well, something has to be there. There has to be some door that allows stuff to get through from one side to the other. And in fact, it exists. It's now very well known, and it is called the tight junctions. The tight junctions, in fact, are made up of this really wonderful series, a conjunction of proteins that really are interconnected. And there is occludin and claudin and Zoe 1, 2, and 3, and it goes on and on and on. And we're now beginning to understand that this conglomeration of proteinaceous substances is sitting and is like a junction between the cells. And something controls them. It is able to tell them when they should decouple or allow an opening, and when they can come back together again to close the door. Because of course you got to close the door again. You don't want it to be open all the time. That causes its own problems. And so What was discovered about 20 years ago was that there are a series of peptides, small proteins, if we can call it, that are the controllers of this. And the best known of these is zonulin. It was discovered by Alessio Fasano and his team in Maryland. Alessio, who's become someone that I consider a friend, he and his team were able to discover, for reasons which we can get into if we have a lot of time, that zonulin in fact exists, and that the intestinal cells, that one cell barrier, has receptors that are able to glomp on to various types of bacteria or food, and that stimulates the cell to produce zonulin. And zonulin, in a bit of a circuitous path, eventually ends up saying to those tight junctions, open. And when it does that, they open, and they open for a few seconds. They can open for a few minutes in some cases, which is a long period of time. And then when it stops stimulating them, they close again. And so zonulin, which in humans is actually a pre-haptoglobin 2. I'm not going to go into too much detail, but it's in fact synonymous with zonulin that is seen in various other animals and so on. So it is a well-known entity, and it is the first objective biomarker that we have found that we know controls this space between the cells, this critical opening. And that, in fact, not only opened up the trapdoor, but opened up our minds to really understanding that the gut is a very dynamic system that is able to push stuff back and forth between the rest of the body and the gut. And that opened not only research into how that functions, but gave us a much more fundamental understanding of how the gut affects the rest of the body. And you mentioned autoimmune disease. There are many other things which we can go into. So zonulin, to be clear, and another small group of peptides related to it, we call them the zonulin family of peptides, are the controllers of not only the cells of the gut, but we've now found zonulin and these other little peptides in cells all over the body, including the heart and the lungs and the kidneys and the blood-brain barrier. And I don't think it takes much to say, wait a minute, does that mean now that we have a way of understanding how the body can control what gets into the area of the brain and what it manages to keep out? And the answer is, yeah, that's exactly what's going on.
Wow.
So it's opening up very exciting pathways for research.
That's incredible. And what a discovery, right? A new breakthrough with regards to how that all affects the body in many different areas, right? And
absolutely.
So let's go back just a little bit, because we do have, I want to kind of touch on this, because you had talked about the discovery and just of zonulin and how that came about. Share a little bit on that, because I'm curious.
Well, it's fascinating, because some of the original research that was done by Alessio Fasano and his large group was on a disease which many of your listeners will know very well, and it's called cholera. And cholera is a disease that still is killing hundreds of thousands of people each year, tragically, many of them children. And cholera, for those who know anything about it, is caused by a bacterium, which it's called cholerae, bacterial cholerae. And this bacterium does something very insidious. What it does is that it stimulates the cell of the intestine to produce zonulin.
Oh,
And what it does and that's its way of being able to get into the body, the
right.
cholera bacteria gets in, it is stimulating the body then to produce zonulin. And when it opens, when those doors open, remember the
Mhm.
tight
Yeah.
junctions, what happens is that liquid begins to leave the body because the body is fighting with all its might to get rid of the bacteria and toxin, because cholera produces a toxin that actually stimulates the production of zonulin. And so the
Mm,
body says, can't have that. I got to get rid of it. And it starts pushing liquid out through the body into the gut because those tight junctions are open
open.
and the liquid doesn't stop leaving the body. And
Wow.
that's why cholera, for those who know about it, people die because of dehydration. They
Oh
are
my
vomiting
gosh.
and they have diarrhea that doesn't stop. Diarrhea is literally that liquid coming out of the body through the tight junctions and never stops because the toxin keeps that door open. And
Mm,
remember, it normally opens and closes. But if
yeah.
it stays open all the time, then you've got a problem. So just by luck, Fasano and his group said, wait a second, what is this toxin?
Hmm.
And it was, in fact, the zonula toxin. And they called the peptide zonulin. And that's why we have the word zonulin.
Wow.
It is not because of any other reason than they wanted to give it that name. Given the light bulb moment was to say, well, wait a second. If the bacterium is doing that with a toxin, there is this entity sitting in our body that can be stimulated to open up tight junctions. Does the body have its own internal ability to do that? It must, because why would the tight junctions be there?
Yeah.
And so they discovered on chromosome 16 of our bodies that, in fact, there are genes that produce a pre-haptoglobin 2 molecule, which is identical to the zonulin molecules seen in other mammals. And it turns out that that is what we have in our human bodies. That is the molecule which we are calling zonulin. And
Ah.
there are a couple of other molecules which are very close to it, which do the same thing.
Hmm.
And so the body now, they were able to discover, and that was the aha moment when they said, so that's what's going on.
Wow.
And receptors on the intestinal cells are able to interact with the gut in such a way that they know when zonulin needs to be produced, when the tight junctions need to open, when it needs to close again, etc. And suddenly we understood the fundamental dynamic aspect of the gut. And of course, that opened up the floodgates, so to speak,
Yeah.
to our thinking about how the gut actually functions.
Yes. And so I do want to get into autoimmune because we're kind of moving right into that path. And when we're talking about tight junctions, the first step that a lot of people, especially children now, are dealing with are food sensitivities. And I think you made a very valid point when I was doing my research was that food sensitivities, people kind of group them all into one area, right? In one group. And that's not really the case because there's different small little pockets of food sensitivities and what they actually mean, right? We've got food allergies, we've got sensitivities. And so let's talk a little bit about food sensitivities. And then I want to move into autoimmune and that modulation.
Well, I think the first thing you need to remember is that there's a difference between the term sensitivity and allergy.
Yeah.
And quite often those are used interchangeably. They shouldn't be. We have many different types of immunoglobulins. Those are other proteins which our immune system, which cells in our immune system, the defenders of the body, will send out as both signaling messengers and actually to act as guards. as part of an army of guards that we have in the gut, we have it in all other parts of the body. And one can break those up into four major groups. And I'm not going to get into the weeds, but
Yeah.
I will tell you that we have immunoglobulin G, M, like mother, and A. And those are the immunoglobulins which you can bring together under the word of sensitivity. They are the immunoglobulins which, when they do what they do, and we can get into that in a moment, will signal to the body that there is something that the body needs to deal with to either neutralize, get rid of, or whatever.
Yeah.
However, those are not very high spiking reactions to the extent that they will be incredibly detrimental to the body within seconds or minutes. That is done by one other immunoglobulin group, and that is IgE. And IgE creates what we classically call allergic response. An allergic response is the body saying, oh my, there is something very critical going on here, which I can't allow. And I'm going to send out the big army very quickly to try to deal with this. And that can cause extremely strong reactions in the body. The end point of that being what some of your listeners will remember as anaphylactic shock, which literally is such a strong reaction of the body that it will close down the body's functioning. That's how strong the reaction actually is. That's allergic response. We are not dealing with that when we are dealing with what most foods will produce. And I don't want to throw a screw in the work and tell you, yes, some foods in some people at some moments can actually trigger an allergic response.
Yeah.
That is true. And so some people need to have EpiPens and other things with them because they know that if they take in certain types of food, it'll get out of the sensitivity range and IgE is going to take over, and that's bad.
Yeah.
And then they've got an allergic response. So the vast majority of responses to foods are sensitivity responses. And one needs to understand that most of the way in which sensitivity responses occur is right within the gut itself. The gut is actually a very, as I said, a very dynamic area. And we now know it's not just a tube that has a bunch of bacteria lying around, but in fact is a very complex system that has on the surfaces where those cells are, there's a big covering of mucus. And in fact, the mucus isn't just one layer, it's two layers. It's a layer that, and each layer has its own city of bacteria and all kinds of other colonies that are set up in a very hierarchical system. And every group of bacteria and so on know exactly what they need to do. They know where they need to be. They are not just allowed to move wherever they want. They have to stay within a certain area. And the body is constantly monitoring that by sending little pods through the cells and between cells that are checking out what is going on. These are called dendritic cells, and they actually are checking to see, is there some food in here that shouldn't be here? Are there any toxins here that we need to neutralize? And if yes, it calls in the troops. And those troops are part of what's called secretory IgA. Remember I mentioned immunoglobulin A? There is a subset of that grouping that is taking place in the gut, in saliva, and so on. And it's called secretory IgA. And these immunoglobulins literally live in that mucus. And their job is to sit around looking for stuff that shouldn't be there. And if it shouldn't be there, it glomps onto it. And it says, gotcha. And as soon as it does that, it says to cells that are sitting there, I am now complexed with this piece of food, this bacterium, this complex of an antibody and an antigen, and an antigen is whatever this food particle or bacteria is, and now it gets complexed. And the body says, great, that's what I'm looking for. I'm looking for those complexes. As soon as I've got them, I will neutralize them. And it tries to do that within the gut. It says, you are not coming into the rest of the body. We now have caught you, and we're going to get rid of you. But in order for any of that to happen, you need to have that mucus city. That city needs to be filled with mucus. If there is no mucus, then all of it dies away. It can't function. And so what we're talking about with food sensitivity is, what is the ability of the body to be able to see all of this food coming in? How does it decide what is good, what is bad, what it needs to get rid of immediately, what it needs to break down, in which way does it break them down, and so on? And so what we really look at when we're looking at food sensitivities is several things. Number one, we're looking at the different food groups, right? Classically, we have 5 food groups. Some call them 7. Let's forget whether it's 5 or 7. Let's call it 5. And these different food groups are dealt with in different parts of the gastrointestinal system. And so in the stomach, you got to break them down. You've got an acidic environment. You start breaking down mostly proteins. And then in the intestine, you're breaking down mostly the carbohydrates and the fats. And that continues down to the end of one portion of the intestine, etc. So what you really want to know when you're doing food sensitivity testing, what you really want to know is, how well are the body's enzymes functioning? Because many of the food sensitivities that we think we have are because the body is not properly breaking down those foods. So if you're not breaking down proteins well enough, then what you need to say to yourself is, well, where are proteins normally broken down? Most of them are broken down in the stomach. Oh, maybe I've got a problem with an imbalance of my proteinaceous enzymes. Well, I'm not breaking down carbohydrates well. I have a problem with grain breakdown. The grains. Well, what breaks down grains? Well, mostly enzymes that are coming from the pancreas. Well, wait a second, then maybe I should be looking at that plethora, that grouping of enzymes that emanate from the pancreas. And it's the same with fats. If that's happening, what are the enzymes? Well, the enzymes that come from bile, right? Including bile itself, the bile acids. So if we're having a problem with fats, which you're seeing because the food sensitivities that we're doing on these tests seems to be going up, well, quite often it's because you're not breaking down the fats properly. Well, why am I not breaking them down? Because you have a deficiency, an imbalance of the various enzymes, etc., coming from bile. So when you're doing testing for food sensitivity, the key is not, oh, look at this, there's one food here that's high. Yeah, that may be interesting. What's really interesting, though, are there certain food groups that are showing high? Because that tells you where is the major enzymatic imbalance. And that is key to understanding where food sensitivity and its testing sits within the whole realm. And I should answer one other thing before you ask the question. Well, what if someone says, well, I did food sensitivity testing and I don't understand it. They checked me out. I had some problems with some foods. I took them out of my diet and I still don't feel well. Something's wrong. Yeah, well, what's wrong is that it wasn't just your foods, it was many other things that were causing a problem, and it's not all about the food itself. It's about much more, which of course we can discuss.
Yeah, and I think you're right. I think people try and blame it on foods, and then they do, you know. And I've never been a big fan of going to the first level. It's what's the root cause? Let's keep digging, right? Let's keep digging down that hole until we actually get to the bottom, so that we can fix everything above. And I think that's where sometimes that the testing, especially when people are looking at it without the help maybe of a practitioner, that they're really not kind of digging deeper into that, into what you've already evaluated on, which is very valuable information. And so now, you know,
Absolutely.
let's talk about leaky gut syndrome, or increased intestinal permeability. We've talked about tight junctions, how they work. And so what happens when now we start to see leaky gut? Because that now can lead us down the road of other more serious, you know, kind of disease, if you may, celiac, irritable bowel syndrome, things like that. Let's talk about that.
Well, remember, in order to understand the importance of what leaky gut is, and as you said, it's really leaky gut is a term we use for the intestinal permeability increasing, those tight junctions opening more often for longer periods, etc.
Mm-hmm.
And the question then becomes, well, what happens when it opens up? Well, what happens when it opens up is stuff gets into the body that should never have allowed to get into the body.
Exactly.
And what is that? Well, there are two major things. One of them is macromolecules. In other words, pieces of food that are very large that should never have gotten in. They needed to be broken down much more before they got in. But whoops, they managed to get through that trap door. And now they're sitting on the other side of the cell where they shouldn't be.
Right.
And the other thing that gets in are bacteria that shouldn't be in there. And if you have pathogenic bacteria in much greater numbers in the gut, then those bacteria have on their membranes something called LPS. Those are lipopolysaccharides, which I'm not going to go into in great detail. But here's the interesting thing. When a bacterium is broken down, it sheds that membrane. And when the membrane breaks down, those lipopolysaccharides become free, and they act as toxins. And they actually get in through those tight junction trap doors. Now, what happens when they get to the other side? Well, the other thing you need to remember is that the body considers the intestine so critical to its proper balance and hemostasis, or normal functioning of the body, that fully 70% or more of the immune system of the body, you know, what fights when something comes in, sits literally in intimate contact with the intestine. And I mean intimate. In fact, what's called the Peyer's patches or Peyer's patches are areas filled chock-a-block with immune system cells, all the different parts of what we call our innate immune system, our adaptive immune system, and all of those cells are sitting waiting for action. If those macromolecules, those toxins get in, it stimulates the immune system, and the immune system, depending on how it's been stimulated, how much it's been stimulated, etc., then sets off an immune response. And the key to that is, well, you say, well, so what's wrong with that? That's what it's supposed to do. Well, it is supposed to do it, but it's supposed to do it in a very regulated fashion, which normally means that the threshold for it going into action normally needs to be very high. The body says, don't bother me unless things get really bad.
Yeah.
When things are getting really bad, it's because all those tight junctions opened, all this stuff is getting in, the body is now being flooded with information. That threshold has come down. In other words, the immune system is working overtime and at a time when it should normally not even been working.
Right.
And so it is setting out the big guns. The howitzers are shooting off their shells at a time when they should be quiet. So you have what's called immune dysregulation. The immune system is working, but not in this exquisitely regulated way.
Yeah.
And when it does that, it is sending signals to all parts of the body. You know, they say what's in Las Vegas doesn't stay in Las Vegas. In other words, whatever was in the gut doesn't stay in that area.
Yeah.
Those signals now get sent out to all the different body parts. And depending on your genetic makeup, depending on environmental triggers, depending on your microbiome and how dysbiotic it is, meaning what is the balance of good and bad bacteria, all of that together is going to determine which part of the body is going to be affected more or less.
Right.
Give you an example. Celiacs, which have a tremendous amount of receptors on the intestinal cells to zonulin, will create massive responses to alpha-gliadin, which is in wheat, which for most of us is something we don't really respond to in any massive way. The body may not like it, but it says it's okay. Not with celiacs. So about 3% of the population will have this kind of an autoimmune response. Then there are 10 times the amount of celiacs that have the same symptoms, but they're called non-celiac sensitivity reactions. So that brings you into 20 or 30% of the population. Then you have diabetes, type 1. We used to call it juvenile diabetes. It's now been pretty well established that diabetes type 1 is in fact an autoimmune disease that has its gestation in the reaction of the gut and its immune system, which, as it turns out, targets the pancreatic area, leading us to what we know as diabetes. And then the list is immense. Various types of frank problems within the gut, what we call IBD, or inflammatory bowel disease, we now know has a very, very significant component of autoimmunity. We're not quite sure what all the players are, but we do know that that is another one. Then you have many neurological problems, including, and I'm even going to mention autism as one of the things which is now understood to have as a major vector, something which is impacting the body, leading to autistic or autistic-like responses of the body that have a strong gut component. And this list is getting bigger and bigger by the day.